one of the most striking findings in genetics is that we humans subsist on a skinny, low-diversity branch, while nearly all our relatives amongst the great apes are bushy, diverse clades. Something clearly happened in the clade leading to us that pruned our genetic tree to near extinction. What accounts for this? We'll explore this question in the next two videos. Now, the contrast between us and our closest ape relatives, the chimpanzees, cannot be more dramatic. The chimpanzees presently have 4 or 5 major branches in their genetic tree. Each of the Western, Eastern, and Nigerian chimpanzees, as well as the bonobos, individually have more genetic diversity than all of humanity. The gorillas are like this too. Eastern, Western, and mountain gorillas have tiny living populations, but they are genetically vastly more diverse than us. The genetic tree of gorillas looks like kind of a winter version of the chimpanzee tree without all its leaves. But the basic branches in both chimps and gorillas speak to much genetic variety. We even get a flavor of the former diversity of orangutans, with their very distantly related Sumatran and Bornean members. So why are the great apes so genetically diverse and we're not? Some of our low diversity is easy to figure out. First, nearly all of us are genetically very closely related because our ancestors left Africa and then exploded in population. Indeed, nearly all the relatively paltry genetic diversity of humans is contained in the African lineages who never left the continent. The point is that the problem is even worse than you might have thought. Even our most diverse parts as a species are still paltry compared to the great apes. We have two possibilities to understand this tree. First, it may be that there is some process that has favored the production of diversity amongst apes like gorillas and orangutans, but did not work in us. These apes never had big populations, but they have been isolated a long time on their different islands or mountain habitats, and so they're genetically diverse but not particularly populous. An alternative is that there has been a great deal of extinction that has served to help define the main branches of the genetic tree of the great apes. For instance, the bare branches of the gorilla tree are consistent with either undersampling or a huge loss of diversity in gorillas. The same may be true for us too, since when we toss the fossil genetics of Neanderthals into our clade, we can see that Neanderthals were genetically pretty different from any living human. And, as we shall see, it turns out there's lots of fossils of our lineage of hominins. We are not lacking for diversity, it turns out, but it was nearly all in the past, with only us as living representatives. We can glimpse our past diversity by considering recently extinct human clades, namely the Neanderthals, extinct for about 40,000 years in Europe, and the Denisovians that lived until about 30,000 years ago in Asia. There are other near-recent human groups too, such as Homo floresiensis, the so-called hobbits, who lived on the island of Flores in Indonesia until, get this, 17,000 years ago, and Homo luzonensis, known from 67,000 years ago in the Philippines. We have genetic data only for the first two, but they are as different from Homo sapiens as Nigerian and Western chimpanzees are from each other. Beyond this fairly young diversity of human-like relatives, our fossil record currently recognizes about 10 Australopithecus species and more than 9 species of Homo, nearly all of which are present in the last 3 to 4 million years. Our nearest living relatives are the eastern chimpanzees in the genus Pan, and molecular clocks say we separated from them about 6 to 7 million years ago. Unfortunately, before 4 million years ago, the fossil record of ancestors of Australopithecus becomes remarkably poor. Still, the conclusion is hard to escape that we once had a lot of genetic variety in our clade, but it's nearly all extinct. To be sure, some of the human ancestor diversity may not stand up to scrutiny as more fossils are found or more genetic data is generated. Still, while gorillas and orangutans have had their genetic bush pruned a good deal, ours has been totally decimated. When did that happen? We can gain some clues to our near demise by putting human ancestors on a timeline. Here we see the oxygen isotope record of the past 5.5 million years. Note particularly the increase in fluctuation of the isotope record in the last 1.5 to 2.5 million years ago. That fluctuation reflects the oscillation between glacial climates and interglacial climates similar to today. During full glacial stages, big ice sheets grew on North America, Greenland, Northern Europe, and Siberia. Before about 3 million years ago, the glacial-interglacial cycle was small, and oxygen isotopes were similar to today, reflecting a warm, fairly stable climate. When we place events in human evolution on this climate diagram, the oldest event is our evolutionary separation from chimpanzees 6 to 7 million years ago. This date is not well calibrated and has a permissible range from 6 to 13 million years based on molecular clocks. The estimate turns out to be uncertain because of problems estimating the rate of mutation. Next, we have at least 6 species of Australopithecus that appear between 4.5 and 2.5 million years ago. The Australopithecines had a combination of bipedal and arboreal habitat and small stature, perhaps say 3 feet high. A bit younger are 3 species of Paranthropus, the so-called robust Australopithecines, that range between 2.5 million years and 1 million years ago. These appear about the time of the switch to a cooler, drier climate in East Africa and a turnover of mammalian faunas with the coming of savanna grasslands. Overlapping the robust Australopithecines are the first members of the genus Homo, namely a Homo habilis and Homo rudolfensis with dates of 2.8 to 1.6 million years ago. These fossils have a variety of features linking them with sort of more, frankly, with the Australopithecus group than with Homo, such as having small body size, partly arboreal habitat, small brain size, and large teeth relative to their body mass. Still younger times, there are at least 9 named species of Homo, with the youngest members, besides Homo sapiens, having been extinct for only about 17 to 30,000 years. The clear implication is that our clade is not so bare bones after all, at least not until very recently. We almost certainly do not have a good fossil record of our distant ancestors near our split with chimpanzees, since we have practically no fossils from that time period. This absence of an early fossil record is perhaps not that surprising, since we have no chimpanzee fossils either, suggesting that this evolutionary split occurred in environments like mountain jungles, for example, where the fossil record is particularly poor. The changing preservation state of human ancestors is consistent with the idea that our fossil record improves as the ecology of our ancestors changed, first from the mountain habitats of living chimpanzees to the savanna woodlands and grasslands of the Australopithecines, Paranthropus, and the first members of Homo. 1 million years ago, there were numerous overlapping hominins in the savannas, suggesting much ecological specialization of our ancestors. Human ancestors were diverse right up to 17,000 years ago on islands and other remote places. These people had fire and often sophisticated toolkits. Hence, the present low diversity of the human clade is due to us in the near recent, with people with advanced technologies and perhaps the cultural skills and food provisioning to outcompete our older neighbors.